All right, welcome to The Explainer. Today we're going to crack open the case file on what is, seriously, one of science's greatest detective stories, the century-long hunt for the blueprint of life itself. So you got to picture this. For the longest time, scientists were pretty much stumped. They could see that traits, like eye color or height, were passed down from parents to kids. But the actual molecule carrying all those instructions, it was a complete black box. So where does our investigation kick off? Well, it's not in some fancy high-tech lab. Nope. We're heading to a quiet monastery garden back in the 1860s, where the very first cryptic clues about our genetic code started to pop up. So you've got this monk, Gregor Mendel, and for 8 straight years, he's just meticulously crossbreeding pea plants. And what he finds is what he calls these characters, these invisible factors that control traits and get passed down in these really predictable patterns. He had basically figured out the fundamental rules of heredity. But the thing is, the world just wasn't ready for it. His work was pretty much ignored for decades. Okay, so this timeline here, this is basically our roadmap for the whole investigation. You can see how we're going to follow the breadcrumbs from Mendel's big discovery in '66, through finding some mysterious transforming principle, all the way to the final, definitive proof in 1952 that just slammed the case shut. All right, so the investigation shifts gears. We're moving out of the garden and literally into the cell itself, zooming in on the nucleus. Inside, they found 2 potential culprits for carrying life's code. But one of them seemed so obviously the right answer that it ended up being a massive red herring, throwing everyone off the scent for decades. So just 3 years after Mendel's work, a Swiss scientist, Friedrich Maescher, finds this weird phosphorus-heavy substance. And get this, he got his samples by washing used pus-filled bandages from a clinic nearby. Yeah, you heard that right. He called it nuclein, but he had no idea he just stumbled upon the secret of life itself. Okay, fast forward to the early 1900s. Now scientists have better microscopes, and they're watching cells divide. And they see these little thread-like things, chromosomes, that are behaving exactly like Mendel's characters did. So the thinking was, okay, the genetic material, whatever it is, has got to be on these things. And this just goes to show you, sometimes the biggest breakthroughs come from the most unexpected places. You've got this undergraduate kid, Alfred Sturtevant, and one night, instead of doing his homework, he has this flash of insight. He just starts analyzing lab data, and boom, creates the very first map showing that genes are laid out in a straight line on chromosomes. Incredible. So the case now has 2 prime suspects, right? Both of them are hanging out on these chromosomes, protein and DNA. And look, pretty much everybody, and I mean everybody, was betting on protein. It just made sense. Life is insanely complex, and protein, with its 23 different amino acid building blocks, could create that complexity. DNA, with its measly 4 building blocks, seemed way too simple. It was the obvious underdog, totally dismissed. And then, in 1928, everything changes. A new clue surfaces from a totally unexpected place, a clue that just didn't add up if protein was the answer. It was a real twist in the case. So this guy, Frederick Griffith, is working with 2 strains of pneumonia bacteria. You have the S strain, which is lethal, and the R strain, which is totally harmless. Now, if you kill the deadly S strain with heat and inject it, the mouse is fine. Makes sense. But here's the crazy part. When he mixed that dead S strain with the live, harmless R strain, the mouse died. Whoa. Somehow, something from the dead bacteria had transformed the harmless cells into killers. Okay, so this was a massive clue. What on earth was this mystery substance, this transforming principle? I mean, think about it. It survives being boiled, which would destroy most proteins, and it can permanently rewrite the genetic rulebook of another living cell. The plot definitely thickens. This leads to some really slick detective work in 1944. These 3 scientists, Avery, MacCarty, and Mcleod, they basically did a process of elimination. They took the extract from the dead bacteria and used enzymes to destroy one suspect at a time. When they destroyed protein, transformation still worked. Destroyed RNA, still worked. But, and here's the kicker, the second they used an enzyme to chop up the DNA, the transformation just stopped, dead in its tracks. You'd think that would be case closed, right? The evidence points right at DNA. But no, the bias for protein was just so strong that a ton of scientists just wouldn't buy it. They claimed the DNA sample must have been dirty, contaminated with some protein, so the case was far from over. So believe it or not, the scientific jury is still out. It's going to take one last absolutely definitive experiment. In 1952, this one involving a kitchen blender of all things and some radioactive viruses, to finally settle this thing once and for all. So Alfred Hershey and Martha Chase come up with this just brilliant experiment. They take these viruses that infect bacteria, they're called phages. In one batch, they tag the virus's protein shell with radioactive sulfur, since protein has sulfur but DNA doesn't. In another batch, they tag the DNA inside with radioactive phosphorus. So now both of our suspects have a tracking device on them. Super clever. And here it is, the smoking gun. They let the viruses do their thing and infect the bacteria. Then they literally use a blender to knock the viruses off the outside. And what do they find? All the radioactive protein, it's on the outside of the bacteria. It never went in. But the radioactive DNA, that's what got inside the cells. That's what was running the show and making new viruses. And that was it. The undeniable, irrefutable proof. The DNA was the stuff that got inside and carried the instructions. After nearly a century of clues, red herrings, and just genius experiments, the case was finally closed. DNA, the simple little molecule nobody gave a chance, was the blueprint of life. So, the case of the genetic code was closed. But this whole story leaves us with this final little twist, because decades later, scientists discovered something called prions. These are infectious, misfolded proteins. It's a great reminder that in science, the prime suspect in one case might just be the culprit in the next. Even when a case seems closed, there's always a new mystery waiting to be solved.